The Cancer Institute has really a premier place among cancer centers. What we bring is the best research, really with application. We are applying our research in both basic science and population-based science to the patient.
In biomedicine, we're faced all the time with intractable problems, and cancer is one of these problems that are very difficult to solve. Often, these problems are solved by thinking about problems from a completely different perspective, and that's the kind of attitude and approaches that we foster at Stanford.
Being in a position to be able to make patients better and be there with them as they travel through that journey is a really powerful thing to be able to do every day.
Stanford has really all of the elements to be an intellectual leader. We need incredible science and an incredible understanding of the immune system, and so it's a natural fit to the scientific community that's here. The most exciting science typically happens when groups from different disciplines or with different points of views come together.
We have pioneers in the research for immunotherapy, molecular targeting of cancer, the genetics of cancer, and we have pioneers in radiation therapy and technology and the biological interactions of all of these different treatments. And it's an extremely rare combination to have strengths across the board in all of those different areas.
You got cells in you, they're still working.
The Stanford Cancer Institute is combining that science with the clinical trial needs in order to translate the research of the laboratories into patient cures.
What's unique about the Stanford Cancer Institute is the people. Our faculty, our staff is committed to making sure that our cancer patients get the best they deserve.
It is an unbelievable collection of faculty members and their trainees who are world-leading in many different areas of cancer research.
It's so inspiring to be at a place like this where there's great scientists, great clinicians, and great communication between them. Stanford is a terrific place for somebody like me that wants to be both involved in laboratory research and patient care, and mostly applying that research to advances in patient care, because it's all right here on this one campus.
Stanford has a very collaborative culture. More than 400 faculty participate in the Cancer Institute. One of our major missions is to promote the collaboration and interaction of all those individuals, and really we've been able to bring all of this expertise to the benefit of our patients.
How have you been feeling?
The NCI designation is something that is much sought after by cancer centers. So we were very pleased in 2007 to be able to be designated as a cancer center. And then in 2016, we got the comprehensive designation.
I think the recognition as a comprehensive cancer center creates a recognition of what Stanford has always done, which is make major contributions to cancer and to our cancer patients.
Precision medicine is often finding the right drug for the right person at the right time. Instead of treating everyone the same way, if I know more about your underlying genetic reasons for your tumor, I can custom tailor a therapy for you, i.e. precision medicine.
Genetics and genomics really are just different terms for the same thing, understanding how DNA affects health and disease, and apply them to our patients.
Patients feel empowered to know more about their treatment and how those treatment decisions are being made, and to have an active role in making those decisions. With these new technologies, we can really hone into specific changes in a specific tumor cell in an individual patient to help guide their decision making.
The STAMP molecular test stands for the Stanford Tumor Actionable Molecular Profile. It's taking a biopsy or a sample of a tumor and doing a very large-scale genomic test on that to molecularly characterize that tumor. So it's looking at whether they have mutations in them or changed in ways that help us understand how we might treat that individual's tumor. And the idea is to delve real deep into the molecular biology and the genetics of those individual tumors and try to come up with a consensus or a recommendation for a novel therapeutic approach. And there's also really interesting technological advances happening here at Stanford to do things like sample DNA from our circulating plasma. So this whole idea of circulating tumor DNA, we think will be quite transformative over the next few years.
What are the samples that we identified? Circulating tumor DNA has really exploded onto the scene in clinical care. It turns out that when there's a tumor in someone's body, there's always some cells within the tumor that are dying, even when we're not treating the patient. And when those cells die, some of their DNA can get released into the blood. That lets us get access to cancer DNA through a simple blood draw. So when you draw blood, you're able to get some of this cancer DNA, but it's a very tiny amount, and so you need very, very sensitive methods to be able to detect it.
It takes a soberingly long time before we know whether something is working, and the idea that we could use DNA to get it that earlier has been quite compelling. That can be very informative, not just for telling whether something is failing, but to help pick what the next therapy should be that might help fix that failure.
After a patient receives surgery or radiation to try to cure their cancer, we think we can use these tests to identify patients who are likely cured versus those patients who still have microscopic cancer cells. And if we can do that, then the patients who we think are cured might be able to skip the chemotherapy that's often given after surgery or radiation. Whereas patients where we have evidence there's still cancer cells in the body, those patients would be ideal candidates for more aggressive treatment. And then lastly, the potentially most far-reaching application would be using circulating tumor DNA tests for early cancer detection or cancer screening. One could envision trying to use these blood tests that look for mutant DNA to identify cancers really early, before they've spread, when there's still a high chance of curing them.
So how many genes have you done this for?
The trainees are the lifeblood of the lab and the science that we do. They come up with the best ideas and the best ways to test them. It's really important to think about that investment for the future for a lot of reasons. If we don't work on training those people to ask the really good questions, we lose an opportunity to change things in healthcare and cancer research.
This opportunity has been great for me. It really presented an opportunity to really work on the cutting edge of cancer research, as well as improve my skill set. The main advantage I've found here at the Cancer Institute of being able to really broadly ask as many questions as possible and getting a nice, helpful answer and being able to move your research forward.
Having the potential to do translational work that can have a very clear clinical impact really is a reason that inspires me to be here every day and gets me excited about being able to impact other people's lives.
As a kid, I grew up and always wanted to be a cancer scientist. It means so much to be at an institution where we focus so much on providing the best care possible to the patients. For me to be involved in any sort of way, it means so much to me.
Many patients, when they're diagnosed with cancer, will be treated by an established set of protocols. That works for many patients, but not for all. Some patients will benefit from being treated by a new experimental therapy that has the potential at being much better than our set of established therapies.
Over decades, we have now figured out what makes a cancer hide from the immune cells, and there are new therapies that have been developed that basically unmask the cancer cell to the immune cells in the body, as well as rev up the immune system to act against the cancer. And a phase 1 clinical trial program is a research program that is focused on evaluating and bringing forward new agents for the treatment of cancer. Phase 1 trial is like the first step as we bring molecules forward from the laboratory when they look promising in all the experiments that we do, and then we say that this is a good drug to test. It requires a multidisciplinary expertise of investigators and staff to accomplish this conduct of a phase 1 trial. And above all of this, the overarching theme is that we need to maintain the highest standards of patient safety.
Good. My diagnosis I got when I was 25 years old. It's called myopericytoma. It was in both of my lungs, stage 4, and it's one of the more difficult cancers to treat. Mine is one of the, if not the rarest form you could have. And it was a pretty grim prognosis at first. I had what they refer to as an innumerable amount of tumors, so many tumors that they really couldn't even put a number on it in both of my lungs. I thought I was done. I thought I was done. Typically, less than 5% make it to 5 years. You know, I hadn't really started a family yet. I got pretty negative, pretty hopeless. I remember the doctor telling me, have hope because there's clinical trials that come out all the time and we're making strides in cancer treatment all the time, especially here, so just have hope. The clinical trial I'm on, basically it targets a gene mutation that I have. It's basically 2 genes that I have that have fused together. The therapy that I take in pill form, it tells these genes not to fuse together, so it's kind of the stop button. And without that growth, they start to die. And now it's down to, you know, a couple. And those couple that are there, they don't even really know if they're tumors anymore or if it's scar tissue. I have kids now, you know, I have 2 8-month-old babies. my son Abraham, my daughter Marlo, my wife, and like everything's great. This hospital, this cancer center is cutting edge. This place has restored my hope. Without this place, I'd likely not be here today.
Cellular immunotherapy is a new way of treating cancer. It's really completely different than anything that you can imagine when you think about giving medicine to treat cancer. We take the cells from a cancer patient and we engineer them. That means we put a gene into the cell so that all the daughter cells also have the genetic program and can make the proteins that are being encoded. When they go back into the patient now, I like to say they've been given superpowers because they now can attack the tumor.
CAR T cells are the first effective targeted cancer cell therapy. A CAR T cell is the most direct genetic method to hijack our own lymphocytes and force them to see cancer as foreign. If we can make a protein that is able to attack only that one molecule, then we've really changed how we're able to manage cancer. We're able then to bring cells that can expand if you need more, cells that can localize to where the cancer is and keep pounding on it. That's what the CAR T cell is. This immune therapy is a paradigm shift.
Cellular immunotherapy is a breakthrough because it's a living therapy. The cells that you are administering can last in these patients for weeks, months, even years. I really believe that we are seeing the proverbial tip of the iceberg. I tell my team that we're at the end of the beginning. This is now laying the groundwork for what these cells can really do.
The cycle of science, moving from the laboratory to early preclinical and then early phase 1 studies, is this critical component of the early clinical trial cycle. Stanford is pioneering the new idea, the new technology. That commitment is a cycle.
All right, so remember, you got cells in you. They're still working.
Immunotherapy in many ways has been a breakthrough in many cancers over the last few years, particularly in melanoma, lung cancer, and renal carcinomas. And we see it now also having efficacy in gynecologic malignancies, including ovarian cancer.
We're looking for combinations of ways of talking to the immune system. Some of them stimulate the immune system, some of them take off the brakes of the immune system, and some of them talk to certain cells of the immune system to get them to recognize cancer and kill it. And so we're always searching for better and better ways to do that.
One of the biggest areas of growth right now is using your own body's immune system to attack the tumor. But we believe for that to succeed, you again have to be able to visualize what your immune system is doing. The imaging sheds light into the process by visualizing what's really going on inside your body. And so we believe what's coming up in the near future is a lot more imaging tools. One of the approaches we use for cancer detection are to develop molecular spies that go into your body looking for cancer. In PET imaging, the spies are radioactive tracers that are injected into your body through a vein, and then they home in on cancer cells, thereby enabling earlier cancer detection as well as improved cancer therapy management. We can make spies that are able to produce signals with sound, able to produce signals with light, able to produce signals that are capable of being basically measured anywhere on the physical spectrum. That means you don't always have to use radioactive spies. We've developed more and more sensitive approaches so that you can detect smaller and smaller numbers of tumor cells. That's leading to the ability to detect cancers earlier. We've been able to also make agents that are both diagnostic and therapeutic. They're called theranostic agents. Theranostic agents will allow us to visualize where prostate cancer might be, where it might have spread, but at the same time treat it. This precision medicine or personalized approach is critically dependent on imaging being the eyes of what goes on with a given therapy. For a patient, that means that they have a much better chance of knowing if that therapy is working, and thereby have a much better chance for a better outcome.
So if you can just get his vitals.
At Stanford, we're constantly developing new ways to make radiation extremely precise and focused so that we can deliver effective doses to kill the tumors while minimizing the damage to the surrounding tissues. Stanford is the birthplace of modern radiation therapy. The linear accelerator technology that's now used around the world, the invention of the CyberKnife, and many of the treatment techniques that we use now for very focused radiation treatment, for breathing control and motion management, those were invented here at Stanford. And many of the first clinical trials of this very focused treatment that we call stereotactic ablative radiotherapy, or SABR, were done here first at Stanford. One of the new areas that we're developing is completely new linear accelerator technology that will allow nearly instantaneous delivery of radiation treatment. We call this technology phaser. And the advantage of super fast treatment is that we can give much more precise treatment.
All right, here we go.
The last 20 years or so has been an amazing time in terms of how much we've learned from the collective experience of looking at cancers and their genetics, and this is a whole global effort.
Terrific.
I think the new technologies for following patients are going to reduce the amount of time patients are on treatment that isn't working and increase their availability of new drugs.
We need to push our limits of what we can do and what we know. The only way to really advance our knowledge is to push forward in laboratories, keep thinking about novel approaches, novel mechanisms. We cannot stand still.
CD22 CAR T cells.
I think this culture of risk-taking is part of our DNA at Stanford. The reason that Stanford has been able to contribute so much and has so much potential for contributing even more is because we look at problems from a new perspective, and that is so important at advancing science and advancing clinical care.
Knowing that every time I pipette, every time I write a script, every time we run something, there could be an impact on somebody else, that's huge to me. Every second for which we sit at our bench, we work towards our larger goal of bringing improved cancer care for patients. And I feel like that's what we work for.